Hello, everyone. This is a presentation I gave to a commission in New Zealand a few weeks ago that I'm just re-recording because I don't think the Zoom call we had actually captured it all, and this is good to have on the record. I actually have a history in New Zealand. I did a semester abroad in New Zealand back in 1994, so very familiar with the country, and I come here with what you may consider an apology from our country, and that much of this COVID nightmare that was imposed upon the people of New Zealand originated from the United States and even from the state that I live in here, Massachusetts. Massachusetts is really ground zero for this in terms of the financial interests that are at play pushing this COVID narrative. We have both Moderna and Pfizer here, and we have many of the PCR companies that helped amplify, if you will, the PCR view of this pandemic. So this information is not going to be easy for you to hear, mainly because the news has been flooded with a completely different narrative on this, and so you're not at fault for not having seen this data. There was a very large billion-dollar campaign here in the United States to inhibit vaccine hesitancy. This was from the Biden administration. You probably saw a few weeks ago, Tulsi Gabbard even mentioned that people who were spreading vaccine hesitancy were put on terrorist watch lists. So this is a heavily censored field. If you haven't seen this information, it's not at your own fault. It is because there are very powerful budgets in play to spin a very, very different story. So this news is going to be very unsettling for you. But the information is true, and I have many of the references and citations in here that can back this up. You can investigate on your own. So, why should you listen to this so-called vaccine terrorist? Well, I do have a long history in this field, and I don't have a history that's really financially tied to this narrative, so I can call balls and strikes pretty fairly. I've had about a 30-year experience in this field. I started off my career at the Human Genome Project at MIT. In fact, that robotic pipeline you see up there in the corner is something that our team engineered for the Human Genome Project to purify plasmids. So we live, ate, and breathe plasmid DNA purification on the Human Genome Project. It's been part of my career is building companies that extract and purify these things from various cells and study the contaminants that are in them that may affect various transfections or biological features, if you will. So after my time on the Human Genome Project, I was a team leader for research and development there. We spun much of this technology out that you see in that upper right picture to a company called Agincourt Biosciences, which was selling these DNA purification tools into the pharmaceutical and biotech industry. And this company was eventually awarded a government grant from Francis Collins for over $27 million to be one of the 5 and only commercial genome centers that were working for the NIH. So we were right there side by side, sequencing various genomes the government was looking for with a variety of other entities like Whitehead, Baylor, the Venter Institute, WashU, and we specialized in sequencing things known as phosmids, which were very low-copy plasmids. So you had to be very good at extracting these phosmids that only existed at a single copy in a given cell. Long history in plasmid isolation and sequencing these things. Over 60,000 citations that I have to the publications I have in the field. We've gotten multiple covers of different journals. So I'm very familiar with the peer review system. And this is another source of what has misled us, is the peer review system is very captured right now. They rely heavily on pharmaceutical advertisement dollars, and much like the mainstream media, they are beholden to some of those advertisement dollars in terms of what papers get selected for publication and which ones don't. So there's gatekeeping going on, which is keeping this narrative afloat, but it is starting to crumble. I'm involved in dozens of patents in the genomics field. I've even invented a DNA sequencer that went to market. The NIH also gave us another $6 million grant for building the solid sequencer. Applied Biosystems came and acquired that company and brought that sequencer to market to compete with Illumina. I've been also involved in building genome centers and in doing clinical sequencing of mitochondrial patients. A long history in this field, but I don't have any revenue or interest in the vaccine space. And so when I see what they're doing with these vaccines, several red flags emerged, and I began speaking about it. Now, probably what I've been asked to speak about here is what we found in the Pfizer vaccines, which are plasmids. Pfizer and Moderna both have their expression vectors still in the vaccines. So there's a paper here that I have the QR code up there so you can quickly get to. It's been heavily downloaded. It's been read by hundreds of thousands of people. And this really started this inquiry into what are the consequences of this residual contaminant that's in the vaccines, and does it have any biological implications for the patients who have been injected with these things. There's another paper from David Speaker, who replicated this work in Canada. This is probably the largest study to date that looked at over 30 vials and found it in every one of them. And this work is now making its way through peer review, and various other people have replicated this around the world. Philip Buchholz replicated this in South Carolina. There's a great testimony from him that's online. Dr. Sin Lee, who is well known for having found DNA contamination in the HPV vaccines, was intrigued by this and also replicated the work using different primers and Sanger sequencing. Brigitte Koning has now replicated this in Germany, and her papers are through peer review. And several federal agencies have now come out and admitted that this is there. Now, they disagree on its clinical implications, and we'll get into that, but there is no question that this DNA is in these vaccines, and that some of this information wasn't disclosed. Another group in Germany, Ulrich Kammerer's lab, actually replicated this work and actually took these plasmids from the vaccines and transfected them into cell lines to see how long they persisted in cell lines. And they persisted for several cycles of cell division. So there's some persistence to this, although the exact persistence is still being studied. It is not going away in 48 hours. So this DNA has these SV40 components that we're going to touch on, and you can detect them in several passages of cells after they've been transfected with these vaccines. And that has certain implications for how long this spike protein may be persisting in various patients' bodies after injection. This has become so easy and routine to do that a group of high schoolers who were doing an internship at the FDA White Oak facility went to measure it as well, and they found it to be 6 to 470-fold over the limit. This is published in the Journal of High School Science. So you don't need a sophisticated laboratory to find this. It's actually quite easy to find, which makes it a little bit more shocking as to how it ended up there and how the regulations have missed this in the process of giving out 13 billion doses to 5 billion people. There's a whole list of studies now that have replicated this. Every one of these studies you see here that's listed in red on the right has found it over the limit. There is one individual who has not published their methods yet that claims it is not over the limit. This person has some ties to Moderna, however, so there are some conflicts involved there. I think you'll find most of the other authors here do not have any financial ties to the vaccine industry and are reporting things with a little bit more consensus as to what the problem is. There is another paper that has emerged as of late, but it does also have some BioNTech ties. There's a paper from Kaiser et al. That claims to have found this to be slightly over the limit, but not as much as everybody else, and deemed it not to be anything of consequence. However, this paper has some flaws in that it used a DNA isolation method that lost a lot of the DNA, particularly the small DNA. It couldn't capture using an ethanol precipitation of that DNA. So that has been refuted by both Kammerer and by Koenig. And the methods that I've reviewed, I think Kammerer and Koenig have this right. They've adequately captured the smaller molecules and are counting those appropriately. Okay, so why do we care about DNA? There is, in fact, a limit. This limit of how much DNA can be in a vaccine was derived many decades ago, and it used to be 1,000-fold higher. 1,000-fold lower of a threshold, in that it was 10 picograms that they would allow an injection. And since they've gotten a liability waiver on these vaccines here in the United States through the National Childhood Vaccine Injury Act, They have dilated and loosened those restrictions by 1,000-fold over the last few decades. But all of these thresholds were based on the assumption of injecting naked DNA, which does get degraded pretty quickly in your bloodstream. You have LNPs, you have DNases that go and actually chew up this DNA, and that limits its half-life to 5 or 10 minutes. The change that's occurred in order to make this mRNA platform work, that mRNA would also degrade very quickly and be a very useless therapeutic if it were not coated in a lipid nanoparticle. And the challenge we have here is these lipid nanoparticles are also coating this contaminated DNA. So the half-lives and the limits that they've put in place are no longer valid. This DNA is going to get delivered directly to cells. It's not going to get degraded. And arguably, the limits they have in place are now completely irrelevant. Even with these limits in place, all of these papers are reporting things log scales over. Orders of magnitude higher than this are what are being seen. 100 nanograms to a microgram of DNA is what is being found in these shots. So the limits are relevant and obsolete, and it's still over that limit by 10 to 100-fold. All right, so we don't know how long things last now that they're in LNPs, because now they're going directly into cells, and that's going to create a much different picture of the persistence of this DNA and its biological impact. So how did this happen? Well, the trials, this is what will shock most people. You have not heard about this in the news, but if you look through the trial work and all the communication back and forth with the regulators, you'll see that Pfizer ran this trial on a different product than what they injected into billions of people. All right, this is a major bait and switch that the public is not aware of. When they made this initial vaccine, they were able to do an additional purification step known as PCR. They would PCR amplify the spike region off of their plasmid that they used to construct this mRNA. They'd amplify it, usually a millionfold above background, and then they would make the RNA off of that. So there was a very small background region of DNA that was used to make the RNA that made these shots. When they had to scale this up, that was too expensive of a step, so they dropped it and skipped it, ripped it out, and went off purifying these plasmids directly out of E. coli without that amplification and purification step. So now what you have is a much higher percentage of background plasmid DNA and probably some of these E. coli components like endotoxin in the shots. We haven't measured the endotoxin, but it's one of these things, if you're in plasmid purification, you know endotoxin from E. coli is your number one enemy when you're trying to purify plasmid of therapeutic grade. If any of that endotoxin gets into the shots, you will create a septic-like anaphylaxis, which we saw many people have post-injection with these vaccines. They would drop in the vaccine centers such that they had to change the administration protocols to have people sit down for 15 minutes after the injection. That is likely, although we don't have evidence and proof of that, that is likely an artifact of these E. coli components known as LPS, lipopolysaccharides, or otherwise known as endotoxin, still being in the shots. Now, what we do have evidence for are these plasmids are in there without any question now. This has been replicated in many places around the world, and there are consequences for having these residual pieces of DNA left inside the vaccines that we're going to touch on. This is not conspiracy theory. This has been published in the BMJ by Retzef Levy, and they go through this process one where they ran these vaccines in the trial and how they changed that when they decided to go and inject the rest of the world. This is traditionally a mortal sin in vaccine manufacturing or in any sort of biologic manufacturing. The process is the product. You change the process, you have to go through trials again, and the EMA even asked them to do that, although they failed to. They asked for another trial of 250 people once they changed the process, and that data was never delivered. So this bait and switch is very important for you to understand why the trial data is of absolute zero consequence to what we're actually seeing in the field. Those numbers are a caricature of what they're actually doing with these injections. So what did we find when we sequenced these vaccines? We noticed you take the DNA sequence that you get and you put it into commercial annotation software to annotate these plasmids. And these commercial annotation software tools, by default, will highlight all the parts in the plasmid that are functional. So there's always an antibiotic resistance gene in these plasmids. That's what helps them propagate in E. coli. You have to grow these E. coli in the presence of antibiotics, and that means only the cells that have the plasmids replicate. But they also highlight things like these SV40 components that are used to drive the antibiotic resistance gene. Now, what's notable with the plasmid map on the left is that the software tool automatically annotates these regions, and somebody at Pfizer had to go and remove those annotations by hand before handing the plasmid map on the right to regulators. All right, so the regulators did get the DNA sequence, but the sponsor is obligated to annotate every open reading frame and every promoter on that plasmid, even if they don't know what they do. And this is where Pfizer got themselves into a bit of a sticky here, in that they had to have gone in there and actively removed those annotations, hid them from the FDA. And the FDA, once they saw our data, were able to go back, look at the sequence, annotate it themselves, of course, years later, to find that, yes, this omission had occurred, and this is a violation of the guidelines that are given. So there has been an intentional deception here that has gone on that I think everyone should take note of. Now, why are they hiding SV40? If you look through the vaccine history here in the literature, you'll know that the SV40 virus contaminated the polio vaccines. And there's a large debate in the literature that's been going on for decades as to its implication in cancer. When you give SV40 viruses to cell lines and to tissues, you will get cancers. There's no question about that. The only debate that rages on is whether or not you can find this in the epidemiological literature. And when you do studies looking at people who got the polio vaccine and who didn't, is there a real difference in cancer rates between those? And that has been much harder to see, predominantly because that vaccine shedded. So the unvaccinated got it from the vaccinated because it was a live entity. All right, so the epidemiological data is always loaded with confounders, makes it very difficult for you to find these signals, but in the laboratory, this virus is one of the most potent oncogenic viruses we know of. So we don't have the whole virus in these vaccines. We have some of the more carcinogenic components of that virus in the vaccine, however, and I'm going to touch on some of the papers that point that out. These sequences are very functional, and they have consequences when they are left behind inside of a vaccine. So the initial public response to this, the regulatory response, was yes, it's there. We weren't informed about it, but the DNA is probably too small in length to matter. And I say probably because they didn't measure it, and they don't know. We do. They then said it's too small in quantity to matter. Well, that quantity metric, as I mentioned before, was never considering there being lipid nanoparticles around, so they don't have their metrics right there. And multiple papers have now come out saying it's actually over their limit, even if that limit is still justifiable. And then they want to claim that they're not functional. Now, all three of these things are wrong, and we're going to go through the literature that demonstrates why they're wrong. First off, that DNA sequence that's in there is known as the SV40 promoter and the enhancer region. That's one of the regions. There's also an SV40 origin of replication that we'll touch on, and there's a SV40 polyase signal. Those are sort of the three main pieces of DNA that are in there. A couple others that are of less significance in my opinion. But the SV40 enhancers, these are used in gene therapy plasmids. All right, these are nuclear targeting sequences. So these transcription factors bind to that sequence and drag it and everything connected to it into the nucleus of the cell. So when you hear a lot of the responses to this work, they'll say, well, it doesn't matter, it's never going to get to the nucleus. There's two things wrong with that statement. One, there's a nuclear targeting sequence in there that guarantees it will get to the nucleus. Second, these LNPs go to cells that are dividing. And when cells divide, there is no nuclear envelope protecting the DNA. They have to go through cell division, and that dissolves during that process. So when they're talking about this not getting to the nucleus, these people are doing a smokescreen on you. They're not being honest with themselves that cells divide. And when cells divide, there is no protection of this DNA. So that's something to watch out for. When you see people saying it doesn't get to the nucleus, that's a sign that they are trying to lie to you. And it doesn't take much more than a high school biology textbook to prove them otherwise. Independent of that, this is a very potent gene therapy tool. In fact, this plasmid was pulled from Pfizer's gene therapy department. That was actually mentioned in one of Pfizer's papers in terms of how they developed these vaccines so quickly. They called up their gene therapy department to get some plasmids they could use quite readily. The other literature out there is that this SV40 promoter and enhancer, this binds to the P53 gene. P53 is the guardian of your genome. It's a tumor suppressor gene. You don't want anything binding to that gene. Otherwise, you may see an impact in cancer. This is very important given the literature we have now on the spike protein itself also inhibiting the expression of this gene. Welfik Elder-Aries group has published a paper, Zhang et al., that touches on that. So now we've got two reasons to be concerned about P53. The DNA in there interferes with it to some degree, and we know the spike protein plays a role in that pathway as well. So there's an oncogenic concern here. There's also listed literature now from Sinickel et al. That demonstrates that this particular sequence is what is known as a hypermutability element. All right, it will induce mutations in DNA near it. So it is, as this paper says, potential tumorigenic activity. So when they say that there is no function to this DNA, you merely need to point to these three papers and say, no, there's published function of this DNA that needs to be explained. And this is not something that you can just say, well, they forgot to tell us about it. No, it's very relevant. There's a reason they forgot to tell you about it, is they didn't want you to know this was in there. And you'll see that this lines up with some of their future business interests. All right, so these FDA guidelines we spoke about earlier, that they're derived injecting naked DNA, but you'll note the papers that justify this limit speak to the fact that the smaller the DNA is on a per nanogram basis, you have many more copies of DNA. So to put this into perspective, 10 picograms of genomic DNA was the limit before. It got raised to 10 nanograms. 10 nanograms of genomic DNA, human DNA, like the genome is 3 billion bases long. It's about 1,000 copies of the human genome. However, 10 nanograms of 200 bases of DNA is 500 billion copies of DNA because you need more pieces. Now, why does that matter? Well, when the DNA is broken up into smaller pieces, it becomes more like buckshot when it goes against your genome. There are more chemically active ends of the DNA. The parts of the DNA that facilitate this integration into the DNA are the ends of the DNA, the 5' phosphates and 3' hydroxyls in the DNA. So the more of those you have, the higher rates of integration you'll see. So when you chop big DNA up into small pieces, its molarity goes up. In other words, a copy number goes up, and there's more of those active ends. And so this is why their own literature from the FDA points this out, that if this DNA gets to viral sizes like plasmids, the limits need to drop down to attograms or femtograms. All right, this is what we have. We have very small broken up DNA that has lots of active ends, and there is a litany of literature out there that demonstrates there's a very high spontaneous integration rate with DNA like this when you transfect it. When you put it in a lipid nanoparticle and put it into a cell, it's like 7 to 10% of the cells get an integration event. All right, and this paper, I think, undermeasures it because it had to integrate a fairly big piece of DNA that had a reporter gene. We don't have reporter genes on these pieces of DNA, so I suspect this paper is undermeasuring the rate of integration because many pieces of DNA in that paper probably integrated without the reporter gene and that they couldn't see them. So this is not something to be brushed aside as a low-frequency event. And you don't need to look at these papers to know this. You can look at Moderna's own patents to know this. They speak to the fact that this DNA is such an integration risk that they need to invent new methods to remove the DNA. And they do a better job than Pfizer. They pull the DNA out and much more effectively than the Pfizer vaccines, approximately tenfold. So you don't need to look to our papers on this or our claims. The manufacturers of these vaccines have patents that speak to this being an oncogenic risk. We also have papers from Strayer et al. That demonstrate SV40 plasmids like this are known to integrate. That's why they use them in gene therapy. Lots of literature from this particular laboratory on SV40 plasmids and integration frequency. So it does lend us to be concerned that this residual DNA can find its way into the nucleus and alter the genome. Now, I didn't put up a paper from Quan et al. It's another one I want to put on the record here. Quan et al. will demonstrate that the sin of getting this DNA into the cell isn't necessarily an integration event. Once you load the cell with cytosolic DNA, you can trigger the C-gas sting pathway, which is oncogenic in and of itself. So really the damage is done on transfection. Whether it integrates into the genome is somewhat irrelevant because this plasmid DNA can replicate episomally, meaning it doesn't need to be in the chromosome to replicate because it has an SV40 origin of replication. That's a mammalian origin of replication. That should never be in a vaccine. You never want a vaccine that you're growing in E. coli to also have the capacity to replicate in the host that you're going to inject it into, mammalians, mammalian cell lines like humans, okay? That is a very reckless and dangerous thing to do. Moderna didn't do it. And there is evidence that Pfizer is at fault here, and that Pfizer did leave this origin of replication behind, and that is only done because they were lazy and careless about this. So why do we care about this so much? Well, there's all of these papers that have rolled out demonstrating nucleic acid persistence. And I say nucleic acid because we don't know from these papers whether they're measuring DNA or RNA. They used a method known as RT-PCR, which amplifies both. And so many of these papers published before this DNA contamination was known, so they assumed it was the RNA because they were looking at the spike sequence. But if they went back and tortured that with perhaps primers that looked at the plasmid backbone that's not supposed to be in the vaccine, I'm kind of curious what they would find. But we have the Krausen paper here that was finding this. What are they? They're out 30 days in heart tissues. There's the Rolchin paper, which is out 60 days. There are all these other papers that found it in placenta, 2 to 10 days. There was found in plasmid from Castroiu's paper, 28 days later. In plasma, and then in breast milk, this was out to about 5 days in the Hanna paper. So, and the Hanna paper is under measuring this. They have a PCR assay that's horribly insensitive. It only has about a 400,000 copy LOD, limit of detection. That should be down at around 10 copies. So their assay, for some reason, is insensitive to this issue. So I think if they had a more sensitive assay, they would have found this out 10, 15 days in breast milk. So anyway, there's a tremendous dose getting through breast milk. If you add up what's in that paper, the number of feedings that a child has, this is not an insignificant amount as those authors suggest, because they did not consider how much milk a baby drinks over the course of 3 days. And that would add up to a substantial and a sizable dose that that child is getting in that feeding. And now, very recently, we're also seeing spike protein persistence in these two papers. Okay, there's the Patterson paper that found this. Have these reversed here. The Yale study actually found it 709 days out, and the Patterson study found it 245 days out. But this is protein that is not going away in 48 hours. It is there almost years later. Why is that? Proteins don't do that. Most proteins have a turnover rate that's in weeks, not years. So this implies something is regenerating the spike protein in your body, or it is evading destruction for long periods of time in certain wet reservoirs in the human body. So this leads people to believe perhaps this mRNA is lasting longer, or there's plasmids in there still generating spike, and we don't know the answer to that yet. Okay, so now here's the real kicker that I haven't presented on much lately because this is very recent work. The kicker here is that we're now finding these sequences not just in the vaccine, but we're finding it in people. And we don't know if it's integrated, but it's there. And these are studies that weren't looking for it, and the methods they used arguably suppressed the signal significantly. All right, so there's at least 5 peer-reviewed studies that have come out looking at RNA sequencing of people who were vaccinated and unvaccinated. And if you dig through that data that's in NCBI, you can take all those reads, map them against the plasmids from the vaccine manufacturers, and you can find that DNA in these patients. There's a study from Ryan et al. That looked at like 75 people in Australia, and there's a great study from Chakrabarty that went through that data and demonstrated both Ryan paper and the ODEC paper have Moderna and Pfizer vaccine sequences in the patient's blood. So this means the blood supply is contaminated. That's a very serious issue if the blood banks aren't looking for it and re-injecting this into other people. This would have normally caused an enormous halt in any other scenario once you find components of the vaccine that were never declared that have these links to oncogenic sequences, and they're now floating around the blood supply and no one's doing anything about it. All right, that should be a concern to everybody in the transfusion field. There's 3 other papers that have recently confirmed this. Now, these papers were not looking for it. The authors were studying other things, but their data has been peer-reviewed and put into the NCBI short read archive so anyone can comb through their data and find these sequences in there. But the Lee study looked at this, the Nabel study looked at this, and the Krausick study looked at this. All of them, I have documented that there are residual sequences from the plasmids that are in those studies infecting their recipients. Some of the studies are actually very useful. One of them was looking at a particular disease that I'll point you to my Substack that goes through these. But this particular disease state, they did RNA sequencing on to see the difference between the vaccinated and unvaccinated. And you can see some differential gene expression that's occurring in the C-gas, sting, and the interferon pathways. All right, that's a sign of potential DNA stimulatory response, okay? So some of the RNA sequencing is actually quite revealing as to what the nature of the contaminant is, because you can see certain gene expressions get turned on or off that are signatures of a C-gas sting-like event that could be induced by the DNA that's there. So anyway, our Substack has that documented, and it's worth having a look. Okay, so I just have these up here so people can quickly get to these papers. I've listed them before, but in case you want their actual decorated titles, here's Chakrabarty on the upper left. He went through the Ryan et al. And the ODEC study, and when we map those reads to the plasmids, we get a significant amount of sequence coming back from spike. That's not surprising because the RNA is certainly sticking around, but less sequence coming from the plasmid DNA, which is not surprising because there is a higher molarity of the RNA in the shot than there is the DNA. And it's not surprising, these methods are using an RNA-seq protocol. These RNA sequencing protocols use RNA purification kits that tend to eliminate or suppress the DNA that's there. All right, so DNA has a harder time getting through here, but we're still seeing that DNA come through. That's what is so shocking about these studies, is that they're employing methods to suppress the DNA, like using a DNase enzyme in their prep, yet it's still there. That means that it must be at much higher concentrations if the researchers or if they were not to perform that step. If they sequenced the DNA of these people directly, they would probably see a tremendous amount more signal. This is the ODEC study, and there you can see in the upper right panel there, that is an IGV screenshot of reads across the plasmid, and the entire vector backbone is fully covered with sequencing reads. So this one is heavily contaminated in these patients. The vaccine DNA is clearly, all regions of it are clearly covered, including the SV40 regions. Those SV40 promoters are making their way into patients, and we can see evidence of this littering the short read archive across multiple studies now. The Nabel study had a much lighter density of reads, so this one, there was less there looking at this, but we can detect some pieces of plasmid DNA that are in there, so another concern. And likewise, the Lee et al. Study has some of these SV40 reads as well. So it's not in every patient, because some of the samples they take are 14 days out or later, but the people, when they take a sample that's pretty close to vaccination, we see it, even though they're using methods to suppress it. Now, this is a study that was actually looking, it wasn't in humans, but it was in mice. It was looking at something completely different. It was looking at the readenylation rate of these vaccines. After you put them into some organisms, the poly A tail gets regenerated, which is another reason why these might be lasting longer. Typically, the length of the poly A dictates how long the RNA sticks around. All right, so they've found some processes in these cells that are readenylating these things, which may be adding to the longevity of the RNA. But the DNA is coming through these as well, so that contamination is actually plaguing that study as well. So why do we care about this? Well, there have been several people and several data sources that have been demonstrating a rise in cancer. All right, there's John Bodwin's work where he FOIA'd the death records of all of the people in Massachusetts. He's done this in many states now. And you can go through those death records and count up how many more neoplasms are we finding. And there is a clear rise in certain types of ICD-10 codes that dictate that this is, in fact, a phenomenon. We have an increased rate of cancer. There's no doubt about that from his literature, from his data. There's others who have looked at this from an excess mortality standpoint. Ethical Skeptic has done this. His analysis does require some important adjustments, in that there is a pull forward effect that he adjusts for, which is during the COVID pandemic. A lot of elderly passed away. And that creates a bit of a shift in the data when that happens. But once that's adjusted for, you can see that there is a clear rise in cancer rates that is persisting even to this day. David Wiseman found this in the CDC data as well. But there are other forms of analyses you can look at from economic indicators. What we're also seeing from Edward Dowd and Ethical Skeptic is that the cancer treatments are up. If you look at the drug sales for cancer rates, this kind of confirms the epidemiological data when you see it in the drug treatment sales moving at the same rate, and that the inflection time point of when that has occurred is concordant with when the vaccine programs rolled out. All right, so cancer is up. The mechanism for this is still in question, but we don't need contaminants in these vaccines that are linked to it. This is also seen in a study called the GIBO study, which was curiously yanked off of a preprint server after I gave a presentation on this study, because the study was quite remarkable in that it looked at excess mortality in Japan. So Japan's a very interesting case study in that Japan is the most heavily mRNA vaccinated country in the world. There are 13% of that population took the 7th shot, and an even higher number, I think it's north of 80%, took the first 2 or 3. All right, so it is there isn't a cleaner picture of this probably than any other country in the world. And what they have seen in Japan is that the excess mortality that's occurred since the vaccination program is larger than the tsunami in Hiroshima combined. This is a massive hit to the demographics of their country. What's also evident in their data, as you can see on the slide or the graph on the right there that has these orange bars, is the magnitude of the excess mortality is staying somewhat consistent as they layer on more doses. All right, that suggests there's a cumulative effect, because what's occurred as they've gone on, you can see the blue wave down below, is less and less people are taking these shots in Japan, yet the magnitude of the excess mortality isn't going down with it. So your 7th shot may have some residuals from the 1st, 2nd, 3rd, 4th, and 5th, right? That cumulative damage means that this wave of excess mortality, they're not out of the woods yet. They may see this persist for a decade. That is quite concerning, given the decay in the uptake of these vaccines over time. The other thing that's very important about this study is that the types of cancer all changed. Prior to this, they had a baseline cancer rate of different cancers. After the vaccine, the cancers shift to a younger age demographic and different types of cancers. That's a very important sort of Bradford Hill tenet of causality, if you will, when you see a biological signal like that. So the GIBO paper is up on our Substack because they pulled it off the preprint server once this came live. But there's another paper from Japan that recently did get through peer review that emulates this, and not to the detail of the GIBO paper, but it does show that this excess mortality in Japan is in fact real, and it's evident in the data. For the first time in their history, they have a decay in their life expectancy. You can see that on the chart over here on the left, where the tsunami and the vaccine are mentioned. You can see their life expectancy was cruising along, getting better and better over the years up until the vaccine program, and now it's decaying for the first time in a very long while. Now, the excess mortality in Japan is not all cancer. In fact, only like 5 to 7% of it are cancers. A lot of the there's a survivor bias going on here. You have to not die from a stroke or myocarditis or acute renal failure in order for them to find cancer. So the people are dying predominantly from these other things. But if they happen to survive that, then you can detect the cancer signal. So it is a bit more delayed and a bit more, I'd say, camouflaged by the other more acute forms of injury from these vaccines. Why is this happening? Well, they know something. Pfizer, very early on, they had the data on this from their trial. They knew this was going to happen, and they quickly went out and acquired cancer companies. They put $43 billion into the acquisition of Seagen, and they put $2.26 billion to acquire Trillium Therapeutics. Trillium was focused on blood cancers that have a CD147 marker on them. Okay, that is one of the markers that is known to be involved in COVID. So they have a very interesting window on those malignancies, and they're buying up the cancer companies that are probably going to play the biggest role in benefiting from the mess that they've created. So in summary, the Pfizer vaccines on the market are not the same formulation as what was tested in the clinical trials. This is a big bait and switch, and it's a fraud. So you can't believe anything they're saying about the vaccine efficiency, which we have seen even those numbers decay over time. This is probably why. They're not really what they trialed. They gave you something different. There is significant DNA contamination. It's found, like 10 out of 11 studies have found this. And the ones that haven't found it have some financial conflicts. So I think the consensus is out. 10 out of 10 of the real studies are finding this. Several are through peer review, which have not been easy to get through peer review. The peer review journals do not like these papers. They get beat around in peer review for months to years, but they're making their way out now. There is also significant DNA contamination now found in 5 peer-reviewed studies that were not looking at this. They were looking at people's blood and tissue, and it was accidentally in there. Other people had to go sleuth it out. We've got cancer on the rise, and there's several papers that report cancer post-vaccination. Like right at the site of injection, they'll see neoplasms. All right, there's something going on here. This can't be ignored, saying it's a coincidence anymore. Now, these are liability-free, and they're often mandated, okay? This may be the largest carcinogenic hit ever to the human population. And we have these on childhood schedules. We're giving these to pregnant women. This has gone absolutely off the rails, and it's time people pull back and look at this data more soberly without the panic of the COVID pandemic clouding everyone's judgment, if you will. Now, we've done some recent work as well I want to point people's attention to. There's another type of artifact in this data that hasn't been covered as well because it's more expensive to cover. So DNA is pretty easy to measure. We have PCR machines that were testing COVID in parking lots. It's a very decentralized tool that anyone can use, which is why the replication has been so easy. What hasn't been easy to replicate and to study are prions and amyloidogenic peptides that we believe are in the spike sequence in these vaccines. The vaccines encode a particular protein sequence in there that's known to be amyloidogenic. That means it can form amyloids or prions in the body. All right. Prions are proteins that misfold, and when they misfold, they chain react to make other proteins misfold. We think this might be playing a role in the clotting cascades, which is why we're seeing lots of clots in people, that these things are facilitating a fibrin clot-like structure, and this is resulting in a lot of the cardiovascular issues that are seen with these very bizarre Kaplan-Meier clots. These are harder to measure. The tools to do this aren't as cheap, and it requires a real specialist. Kevin McKernan has been doing some work, not me, different Kevin, on this, and I've been collaborating with him in that I've been sequencing some of these clots to prove that they're not a fictitious Twitter phenomenon. They're in fact real human DNA in these clots. And there's something that we're noticing in that DNA is that it has a signature of being a neutrophil extracellular trap. It is a type of reaction that happens in sepsis, and your immune system sees foreign entities, it begins to clot around them, and it tends to take some free circulating DNA into those clotting structures and leave a particular signature in that DNA that we can see. This sequencing has been very informative in that we're beginning to see sequences of the patient's genome that might be predisposing them to this. As you know, billions of these shots were given, 5 billion people perhaps. We don't have 5 billion people that are clotted. A subset of those people are having a bad reaction. We're trying to figure out who. It may be that they have genetic predispositions in some of the clotting cascade that make them more at risk of this than other people. And we're beginning to see this right now it's just 2 clots that we've looked at, so we don't have statistical significance that this is in fact a causative mutation. But we are finding what are known as high impact variants inside of genes involved in fibrin formation and fibrin clotting. All right, that is a bit spooky to say the least. So you can see more about that on this particular Substack, and be sure to look at the links in there that point to Kevin McKernan's work. He'll go through demonstrating that some of these clots are binding thioflavin, which is a marker for an amyloid. And so there may be an amyloidogenesis issue that we have. That pathology is going to have to be looked at very carefully because that may be what's behind some of these clotting problems that we're seeing in the field. So Anyway, thank you for the time. I know this isn't pleasant information to hear, but it's real, it's out there, and it is being heavily suppressed. So it's important that communities like yours can hear this, and the citations are in here for your reference. If you have any questions,